And as we begin the program today, I'm delighted to introduce our keynote speaker, Russell Thomson. Russell is an accomplished applied statistician whose career spans more than 2 decades, with a remarkable record of over 100 peer-reviewed publications and extensive contributions to fields like health, marine science, and automated systems. So in his talk, Russell will share highlights from his diverse career, showcasing projects that combine advanced statistical methods with real-world impact. He will also explore the fascinating interactions between statistics and music, offering a unique perspective on the versatility of our field. So please don't forget to add your questions. I'm sure you have plenty for such an interesting talk today on Slack. And then in the end, we'll have a Q&A. And if you don't have time to address all questions, then Russell will be able to see them on Slack and address them later on. So please join me in welcoming Russell Thomson.
Thank you for that introduction. Can you hear me online okay and in the room? Great. I'm just sharing my slides. Yes, that's in the middle, sort of. Okay, so I'm going to talk today about my career in applied statistics. Now, I actually started at Monash University doing a PhD in the mathematics department. My topic was the coalescent process. It was about population genetics. I had this background in probability theory, and I got really excited about probability theory, and it was quite theoretical. And I went on to do some postdocs overseas that were applying some of the work to large populations, but it was still quite theoretical. Yeah, and I thought that I would come back and just work on my own in theory. And then I came back and I visited a colleague, Melanie Barlow, who I'd worked with before at Monash Uni. And I visited her at Walter Eliza Hall Institute of Medical Research, or WEHI, and her boss was Terry Speed. And I spoke to him, and he offered me a part-time position on the spot. And I was quite lucky in that fact, but it gave me an introduction to applied statistics because I started applying my knowledge across a lot of different problems there. And I realized that I actually enjoy working with people on their projects better than I did just working on by myself. I went on to work at here in Tasmania, Menzies Institute of Medical Research and at Institute of Marine and Antarctic Science, working in applied statistics. Yeah, I am talking today to acknowledge country on Munawina land, sovereignty never ceded. And then I went on to do consulting at Western Sydney University. There's a few other things in there, teaching and other stuff, but ideally, or most of my role was working towards being a consultant, statistical consultant, and I was employed at Western Sydney Uni to help PhD students and master's students across all fields. And I started to hear about projects that required statistics in law, humanities, music, psychology, as well as what I was used to, which was medical stats and biology and ecology. I'm now working for Analytical Edge with Joe Potts, who's going to be giving a keynote speaker later today. So the first project I wanted to talk about, so I'm going to talk about some of my favourite projects that I've worked on, basically. I worked on a project when I was here in Tassie at the Menzies Centre on kidney disease in the Tiwi Islands. So this is something that they've noticed is that in the Tiwi Islands, the rate of kidney disease is much higher than even other Aboriginal communities. And so they were thought that maybe there is a genetic link, let's say. So the theory we had was that there is some sort of mutation or variant, genetic variant, that is in the population in rest of Aboriginal Australia that has become more common in the Tiwi Islands since they isolated, that population isolated, say, 7,000 years ago. So there's cultural evidence that they've been isolated for about 7,000 years up until Western influence more in recently. And so the theory is that there's some sort of genetic variant that had little effect on the population until Western influence, and then there's some sort of genetic environment interaction going on that is resulting in this higher rate of kidney disease. So we collected data. Well, I didn't collect the data, but I did actually get to go to the Tiwi Islands and present the results, which was pretty exciting. So the data was collected over a long time. They're a well-studied population, and actually they showed quite an interest when we talked to the Tiwi Land Council about the statistics. They were actually quite knowledgeable, I guess, because they've had people talk to them before. And so it was quite interesting to talk to them about our results. Part of the study was looking at comparing genetically the population with other populations. So these are different colours, are different HapMap populations. These have been used to compare to genetic populations. It's freely available, these genetic data on certain individuals. And so we had a look at how did the Tiwi Island population, our sample, compare to other populations. And we found that the squares are ones that have been marked that they've got different ancestry amongst, and we did see them sticking out as some were closer to the Chinese and Japanese populations and others were closer to the here is CEU is the Western European, and this is Indigenous American population, and the green was Mexico. It was actually the closest, surprisingly, to the Tiwi Island population, interestingly. Over here is the African populations. So we had a few surprises. The other thing we did was we looked at haplotype block size. Now, haplotype block size is due to linkage disequilibrium, something we talk about in genetics. If you think of it into terms of statistics, what it says is how long along a DNA strand do you find correlation. So if you find that the variants or the genotypes correlate with other within a population for longer strands, it's indication that you have some sort of bottleneck in the population, like we saw with the Tiwi Islands, or you have a very small population size. So the interesting thing here was that the Tiwi Islands sat, in terms of their haplotype block size, in terms of the correlation between the genotypes, it stretched for similar amounts to other populations. The smallest haplotype block size were the African populations, showing that they have there's no evidence of a bottleneck. They're a very old population, but there's not that much variant. And this showed us that the Tiwi Islands, even though they did go through a bottleneck and have been isolated, that's not showing in their genotypes, which is possibly because of they have historical kinship rules that help decide who marries who, and that we think are designed to benefit the population in terms of genetics, really, in terms of diversity, and that may have had an effect here. Now, the main point of doing this was to try and find a variant related to kidney disease. And so we did this genome-wide association study. I think others have spoken about this in the last couple of days. In this example, we did go back and replicate, and we found there was, after adjusting for multiple testing, there was only really one or two variants that were significant. And so when we're doing association, normally it's just a simple linear regression or something like that. But here, the population are related because we've got a relatively small population, there are relatives within it, and we had to adjust for the fact that they're not independent samples. You can do that by collecting kinship data as who's related to who. You can do that using the actual genetic data. And yeah, I wrote a book review about all the different methods you're doing that you can do on this. Our favourite method I think we ended up using was something by Tim Thornton, where he actually reversed. You have, instead of having the genotype as the explanatory variable, and disease as your outcome variable, you swap that around, and it makes the stats a bit easier, and it can actually makes it a little bit more powerful. And we ended up using a structural equation model to describe the result, because kidney disease doesn't happen in isolation. There's blood pressure and a lot of other things. So yeah, structural equation models. Also, you could do a Bayesian network model. They're something quite similar, but they're quite nice for describing, you're just almost describing causation when you can't infer causation, right? So that's my first example. Another problem that I worked on, completely different topic, but it was looking at reef life survey data. So this is something from Institute of Marine and Antarctic Science. The researchers, Graham Edgar and Rick Stewart-Smith, have worked together. It's a citizen science project. So they get divers from around the world to collect data, but with the same method everywhere. And it made a really powerful study, data set. And the bit that we worked on was they actually collected also cover data and counted how much coral there were. And we compared it to within a marine park and outside a marine park to see if there's an effect on coral cover. On the marine park had an effect. Here is a picture of the data. I think the reviewers came back and didn't like our description, so they suggested violin plots. So you can see here, these are actually violin plots, although they don't have that nice shape like a violin, do they? But they kind of describe the variation in each site and how many sites we have. So the top one is coral cover, then we have algae cover, and then we have biomass in carnivores and then herbivores. So it's all at the same site. And we also did a structural equation model to try and describe the effect. So we did actually see an effect, an association between marine parks and coral cover, in that there seemed to be more coral cover within the marine parks. And we saw there was a direct effect, but we also saw an indirect effect in that there was an association between marine parks and the herbivores that are more common in the marine parks because they're not fished, and then less algae, which is correlated to the amount of coral. Okay, I'm going to go on to the next subject that I worked on. Thanks yesterday, I think it was it Kunj who gave a nice description of Markov chains, a really good animation. Mine's not nearly as good, but the idea with Markov chains is that you have one state and then looking at the probability of going to another state. Here we've got on the left, rainy, cloudy and sunny. So let's say if it's rainy yesterday, is it more likely to be rainy today? And you can actually test, well, if it was rainy yesterday and the day before, is that going to affect whether it's going to be rainy today? And you can do this testing using multinomial logistic regression. That's what we did in this example of modeling Markov chains. So here, This was actually data from my wife's research project. Her statistical advisor suggested Markov chains, and I thought this sounded like fun, so I actually collaborated with her to work on this project looking at abalone behaviour. So abalone, this is in Tasmania. Abalone is quite a large market. They stick to rocks. You see them, they're like a big mussel with a shell around them. What you don't see is they actually feed sometimes what they call drift feeding, I think, where they stand up and catch the algae in the water stream. But the thing is that they can't do that if they're hiding or sheltered. And they have been known to be affected by this long-spined sea urchin, which is due to climate change. The currents have gone down and the ranges have expanded into Tasmania, and they say they're affecting urchins. Abalone. So what we did was we modelled the behaviour of abalone using a Markov chain. So you can see here on the, get the mouse working. Yep, you can see here that we have 4 states, and mostly they stay in the same state, whether they're exposed, lost, outside the plot, or sheltered. Lost was when we just couldn't find them. My wife actually epoxied little tags on them so you could find the same ones. So we're following individual abalone. And then they added sea urchins into the plot, and we looked at the behaviour, and they seem to go from being exposed mostly to being sheltered now. Or we lost them, or they lost them. And this is week 4, 5, and 6, and you see the behaviour changed a little bit, but they're tending to go now towards the sheltered or lost. They did the same thing with adding abalone, just to make sure that it's not just a species competition, it is about the urchin. And you do see a change in behaviour with abalone, but less so to going to this sheltered state. Cool. I've also worked a lot in machine learning or predictive analytics. When I went to work at Western Sydney University, I sat in a maths department, and most of the people there seemed to be computer science or data analytics rather than statisticians. But they were working on very similar models. So I've just borrowed this slide, but I moved it, instead of a machine, it is a statistical model that you're first testing at the training stage, and then you're using at step 2. How do you tell the difference between a data analyst and a statistician? Well, I guess a data scientist will talk about feature selection, while statisticians will say variable selection. So we do a lot of feature selection, meaning we're choosing which variables are going to affect our outcome. The other thing is you're mostly interested in prediction rather than significance, aren't you, when you're doing machine learning? So I've been involved in a few of these projects. One of the interesting ones was on for this app. It's an air radar app. You can use this and you can monitor your own symptoms. You say, oh, today I am feeling sneezy or stuffed up, or I think there's a few different options, and it'll link your symptoms with the amount of pollen in the air, the temperature, the amount of smoke particles. And what it actually does is it fits a model per individual to work out what features affect your symptoms. So my job here was to look at the model they were using. It was working, but how many symptoms or how many days of symptoms did you need before the model was doing anything? Could we come up with a better model? This is the sort of things we did. And we produced, using R Markdown, 100 reports for the 100 volunteers who wanted to see a whole report on their model, I guess. So yeah, this was a pretty interesting project. It was a consultancy. The interesting thing I found, so the best model for predicting turned out to be like a generalized linear mixed effects model. We used the whole data, and then we had a random effect of individual. So you kind of then looking at sort of saying that there's an overall model and then each individual has some variation in terms, and we used a random slopes model for the variation. While that was the best prediction result, it wasn't really practical because then they're running these models all the time and you can't run it on the whole data because it takes way too long. So they would change the model a little bit because the second model I found was a random forest model. So the random forest model, but it was run on each individual separately. And a random forest is one of these models that use a tree-based, so there's interactions and curvilinear relationships part of the modeling, which is quite nice in an automated setting. Another place that I worked on machine learning, this was a project, it was a large project with a lot of collaborators. I worked more as an advisor in this one, and it was called SIMPACT, which is Smart Irrigation Management for Parks and Cool Towns. What they did was they put a lot of sensors in the ground at Olympic Park in Sydney, and they're trying to create a automated watering system, a watering system. They're trying to predict the soil moisture for tomorrow based on variables today, so that they can set the irrigation. And then the ideal would be to work out the best cooling. So it's supposed to be, when they're talking about cool towns, they've shown that having more greenery cools the earth around cities. And so if we can set up these things, it helps with cooling. So this model, they had a lot of data because they were collecting from these sensors at first it was every 10 minutes or something, and it was just way too much data. And so they had to come back. They realized the sensors were running out of batteries quickly, and it was way more information that you needed. But the things that went into the model were what the irrigation did, forecast precipitation based on BOM, Eurometeorology, humidity and temperature, and then you'd end up with a prediction of soil moisture. Just looking at some of the models that we compared, it turned out that the second best model was just a K nearest neighbor model, which is a very simple thing. We just collect what other data was most similar to what we see there. Let's put that in. Yeah, and so that is the model they used in the end. So with all the fancy different models, you've got some neural networks and some profit. I think that's the Facebook model that's used in here. And then ARIMA is the autoregressive mean average. But yeah, they ended up just we implemented the KNN model. So any of these models you do cross-validation. I just wanted an excuse to include this graphic because I quite like it. You want to do cross-validation, especially when you've got a smaller data set and you want to validate on the largest amount possible. You can do a training based on one less than your whole sample size. Hopefully you've got more than just 8 as your sample size, but it describes it quite well. In this situation, we had time series data, so it was important to make sure that you validate on data beforehand. You can't validate on data that's afterwards. It just doesn't work. This slide was from Rob Heineman's website. I actually was at Monash University when he was there. He is a great time series expert and done some great R packages on time series analysis. So this is just the results from that project. You can see the prediction and the actual soil moisture put on top of each other in that graph, and then the results were put onto a map. Okay, let's go on to another project working on predicting type 2 diabetes. using childhood measures from a cohort study, the young Finns. This is a long-term study in Finland. This is what I'm actually going to be working on next year, one of the consultants for next year. So the data has been collected for many years. So you've got multiple time points on some of these predictors. But there's a lot of missing data. So some of the team have already implemented a machine learning technique that has showed that it can predict type 2 diabetes. And they found the best model was this XGBoost, which incorporates the missingness within the actual model, which is quite nice. But we're still going to work on this in a different way because we actually want to think about it in terms of the clinical benefit of it. So how can we think about if a child comes into the doctor's office and they want to predict type 2 diabetes, what data do we have around them, and how can we present that as information to inform the doctor to give advice? So these other methods, and often in machine learning when you're doing predicting disease or health outcomes, what they use is each time point is just another feature used to predict, and you may impute the missing data, or you may use XGBoost to deal with the missing data. And then you have each time predictor at a different age is a different feature in your model. So what we're thinking about doing is, can we summarize the data for each individual as a life course and use that as a predictor? This is another project I was involved with where Marie-Jeanne Busco worked on the same data, but they're looking at latent class mixture models to class the different people based on their life course data. So over here you had, and here it's BMI, and you're looking at different types of BMI over their life course. So this is something we could use, but this is you're resulting in classes, and we know that using dichotomous data is not useful predicting in machine learning usually. So what we want to do is do something like this, but with continuous data. So we might think about sort of area under the curve of one of these life course models, or we could look at rate of change or something like that, whether they started high and then went lower or something like that, and use these as, and then they would use these to go into a machine learning model. So we're talking about feature extraction, I guess. Another project I was involved in was based around a Department of Education funding. There's these HEP-funded grants, National Priorities Pool. Somebody passed around his email and I read it and I thought this is something we could do. And we ended up getting this grant. And the main reason was because none of the other people who applied for this thought of it in terms of statistical modeling. And so it's good to read around for these because you can get them if other people haven't thought about solving them using statistics. So the project here was looking for an individual-based measure of socioeconomic disadvantage for university students to work out where best to target funding for those with low socioeconomic or with disadvantage. And I led a large team of researchers at Western Sydney Uni, USyd and Wollongong. It was a bit confusing because the idea was that we needed to come up with a better measure, and the current measure was based just on where the students lived. And so there is some problems with that. But then because we didn't have a gold standard of what is disadvantage, we asked a lot of questions, but they were often biased. It was hard to decide what was the outcome in this. So we used the CIFA measure, which is this one based on where they lived. And we showed that, well, it worked worse when you went to somewhere like a remote university to start with, because then the regions were so large, I guess. And we did come up with what was the best ways of measuring disadvantage, but we said it depended on what the university where the students were, basically. So that was the advice we gave. The one best measure was this question: do you contribute financially to support your family? How am I doing for time? I still got a fair amount of time. I'm getting to the more, say, experimental part of the talk. So when I was working at Western Sydney Uni, I found I was doing asked a lot of times about Likert scale data. These are this data where you have from a response is from strongly disagree to strongly agree. And this is my favorite way to describe that data based on a diverging bar plot. So you can see where the middle of the results are, and you can see how everybody's responded. The thing is that often the data I was seeing, people were responding at before and after. So you may want to see how individuals actually changed if they had some sort of intervention. And so I thought about how to visualize this. So this is, if you wanted to visualize it using arrows, I guess. So you could start, those that were strongly agreed, you could see how many went up to disagree or neutral. So that would be one way to describe everybody. But if you had a lot of people, then it would get a bit messy. You could do it using a heat map, was another way I thought about doing it. I think this is actually my favorite way of being able to see. Then you can see if the colors were on one side than the other, then you'd see whether people have moved up or not. This is just a way of visualizing it. If you wanted to test, then you would be testing something like well, if you had enough data, you could treat it as normally distributed, and then you do something like a two-sample t-test, or something using non-parametric statistics. This is actually how with this recommendation, the people end up using these bar charts, which also presents the results. But given my interest in Markov chains, I kind of suggested that this might be a nice way to do it. Now, I did promise that I would talk about music a little bit. So my outside interest is in music, and so I thought about ways that I could put music into this talk. The first one, I just wanted to point out a study on nostalgia where they showed people's interest in music is related to their age and the age of the music, and that there's a peak around 15 or something, that if you're as you get older, you only like music from when you were about 15 or 20. And interestingly, if you're approaching 60 or 70, the music of today is worse than the music from before you were born, according to your opinion. So when people say, oh, the music's no good these days, I think it's more about their age than anything else. The other way I thought about music is in terms of a point process. So this is just drum tablature of the Bo Diddley beat, but just showing that you have a regular beat, then it looks a bit like a point process. Going back to my very first paper I published was on ages of mutations on a coalescent tree. I was working in a point process. But yeah, my interest in music, I'm a drummer, but I'm much more interested in improvised music. So instead of being exactly appearing at the same interval, I'm interested in how those intervals vary a little bit. I do a show on Edge Radio on Sundays at 7 o'clock, which is all about improvised music. And so I've been thinking about that in terms of statistics, I'm always trying to find the within the variation, within the random noise, where is these intervals, where is the signal, you could say. But within music, I'm less interested in the signal, but how much the variation occurs. So I started to play around with something called tidal cycles, which is a live programming language. And I started to play around with some samples. So this was just using tidal cycles. That's the code needed. And this is just a regular drum beat with 8 consecutive drum sounds. What happens if you put some randomness in here? We could just add what beat comes up next. And to me, to my ears, that sounds a bit better. It's not just this common beat. It's varying randomly. But yeah, I thought about, what about varying the interval? So we could add a little bit of code in here, and now the interval is varying randomly. It's just a small amount, so it's maybe not noticeable. I'm curious to know if that sounds better or not. Once you add the interval to see how it starts to approach noise. If you add too much random, right? So to follow on from that, I'd probably think about, well, one thing you could do is like survey and people's opinions about how it sounds. But I thought about it some more, and when you're improvising, you're probably looking about the beat beforehand, so that if it does vary, then you're probably going to match that. And so it shouldn't be this random noise. It maybe should be something more like a random walk, that as you get larger, it should stay larger and then vary up and down from that. But so that's something else to explore. Thank you.
Thank you so much, Russell. A very interesting talk, full of very rich case studies and lots of interesting methods, going from machine learning to classical statistics, and a lot of visualization and interactive things going on. So I didn't see any questions in Slack yet, but I will open for questions in the hubs if anyone in person has a question.
We've got a question here in Hobart from Cameron Patrick.
Thank you.
Thanks, Russell. That was really interesting, and some really diverse projects that you've worked on. So my question is, I guess there's a bit of a trade-off between being a generalist and a specialist in terms of career paths, and sounds like you've gone very far on the generalist path. I'm curious to hear your thoughts about, I guess, how you ended up being such a generalist, and whether there are any, you feel like there are any downsides to that. Obviously, there are huge upsides in terms of all of the fun stuff you've been able to do.
Yeah, well, it depends on what sort of, how you like to work on problems. But I like to think of myself as a problem solver. So I think of, if somebody comes with a data or a problem and then think about a solution, then I often move on to the next one. But if you really want to work through a problem and get every aspect of it, then it's nice to specialize. The other thing I've found, though, that within being a generalist, there is room to say, okay, if this solution needs some new theory, or even theory from a different field that you want to bring across, then you can start getting into your own research where you can concentrate on that for a while, that you don't have to always be flipping between fields all the time. Cool.
Thank you. It seems there's a question in Christchurch.
Hi, Russell. Thanks for the talk. It was really interesting. It's Meredith from Christchurch office, Christchurch hub. I was quite interested in your work in, I guess, measuring disadvantage, and I just have like 2 questions around that. So in terms of capturing disadvantage in uni students, was the only measure you looked at that idea of do you contribute financially to your family, or were there like other, I guess, encompassing factors like income, and other factors? And I guess my second question is, given your work in this, if you had to do this project again, but instead look at measuring disadvantage in children, just like a stab in the dark, what would be the most, I guess, compounding factor in your opinion? Thanks.
Thank you. Yeah, we did look at a lot of other things. We asked about income. Often the problem was trying to distinguish between students that lived at home, because income is often measured as a family income, to distinguish whether they lived from home or whether their income included their situation or their family situation. We asked about financial hardships. We asked about things like what pathways they went through that sort of to get into university. That seemed to be useful. Yeah, ideally we'd have this gold standard of disadvantage that we could use other measures to measure against, but it was hard to find one. Sorry, does that answer your question? There was another aspect.
Yeah. Oh, and just, I guess, unrelated, but if you had to, like, what would be your best guess for the most compounding factor for a measure of childhood disadvantage? Yeah, would you have any guesses for what would be the most influential factor?
Yeah, I think if you could measure something in terms of, it's not just one factor, but things like those that were first in family seem to relate as well. So I think cultural background and or background in general would be good predictors if you could capture a lot of that. Our pie in the sky plan that didn't eventuate was to actually monitor students' time online using the learning software to see if we could see whether they were working, say, at part-time jobs, or whether they can only work on the weekends, or but of course there is a lot of issues with ethics around collecting that sort of data.
Yeah, thank you for that. Appreciate it.
Thank you very much. And there's a question from Perth. I'll go around the hubs this way.
Thanks, Russell, for your interesting talk and sharing your experience and expertise with us. Currently at work, I have a project. It's actually also in kidney disease and also cardiovascular disease. And we're meant to be using structural equation modeling. And I've put that project on the back burner because I don't have experience in that area, and neither do my colleagues. So I'm currently reading Rex Klein's book on structural equation modeling, but I'm wondering if you have any sort of getting started resources for structural equation modeling.
Yeah, I can't think any up the top of my head. It's interesting. It's been used in a lot of different fields. So there's used mostly in psychology to start with, but it can be useful in many different fields. So it's hard to find a reference that is relating to particular profession. So, but yeah, I can, if you want to contact me offline or online, I mean, then I can have a look. I can send you references, suggested references.
Thank you. Any questions from the hubs? Other questions? Or feel free to type in Slack. We have a comment from Xixing in Slack. He said, by the way, I feel very seen by your comment on age and taste of music.
I had a question in Slack too. I wasn't sure if I should.
Sure.
Do you want? You can just read it.
Yes, I can read it. Thank you. Thanks, Russell, for touring us through your amazingly varied career. Can you give some advice on how you switch between such a diverse domain areas? And how do you decide how deeply into the weeds you need to go as a statistician?
That's a good question. Because, yeah, often you can solve these problems with much simpler methods. And so it is worth thinking about your audience or your collaborators in terms of which method you're going to use. So sometimes we think about using the most complex model because it seems the best model, and it probably is. But if it's going to solve the same or come up with the same solutions, then it's not always best to use the most complex model. So I think it's important to work together with your collaborators and work out what their expertise is in and how they can describe the results.